generally am all over the place. This is actually one of the times I'm not talking about CBTN data. I'm going to talk about data we may connect with CBTN and a bit about, hopefully it'll be obvious that you all may be able to tell. So I'm actually taking off my CBTN hat a little bit today, putting on my hat where I was connected to the Molecular Characterization Initiative through a role that I play in the Children's Oncology Group. And so the way that this project was launched through CCDI was in partnership with the Children's Oncology Group. And as all of us know, because I have pointed this out, but you all know this, all CBTN sites are also COG sites, as is almost all sites in the US, Canada, Australia, and New Zealand who treat children with cancer. In the US, it's 90% of children with cancer are treated at COG sites. So that's the role and how I got very involved in the MCI. And then I started having all this deja vu about things we've been working on. Yeah, and what we've been working on in CBTN, which is a lot of the same vision of learning from every child, connecting all this data. So I'm going to sort of talk from the Molecular Characterization Initiative. So the COG, like I said, these are COG sites except for New Zealand, which is like right over here and not pictured. And I like to point that out if you have a global view. Warren kind of explained this part, but basically it relies on the COG to consent patients. And through Project Every Child, which is funded by the COG Foundation, which is essentially a registry for all children. But as most of us know, the COG is National Clinical Trials Network. It's charged with therapeutic trials, large therapeutic phase 3 trials mainly. We don't study most pediatric tumors, and I think you'll see some of that data now because this is the first time that COG has actually really had a connection to the whole rest of the CNS population, and we were very excited to go first as a CNS committee. So what I'm going to present is data that's limited to the CNS subjects who enrolled between when we went live in April 2022 and there are clinical data cutoffs twice a year in the Children's Oncology Group. And so we're using data up to the data cutoff of last December, although the more recent data I think is going up this week. So this is the enrollment, and I actually love this slide. This is the x-axis is actually each line is an institution. And what you see is this is kind of like us, kind of high enrolling academic institutions that see a lot of kids travel to us for therapy. And this is the other half of who is in the Children's Oncology Group. So smaller centers, they may actually not have an academic focus, but we're reaching twice as many kids because we're using this network. And so there were 2170 subjects enrolled in the first 20 months of the MCI. Oh, that's Seattle, by the way. We do consent and enroll everyone on everything we can. This is actually my favorite picture. Yeah, so those kids are on CBTN as well as MCI, I'll point out, which is going to be the next fun part of connecting data. But what I really love about this is that you can see the centers as the big dots here. The little white dots are the kids. So that's the zip code, actually, of where the children live. And part of what I wanted to point out is how we're actually not missing the middle of the country. Kids actually have to travel for cancer care if you live. This is also not geographically where Alaska is, for those of you who don't live in Seattle. But they come to us, and a lot of people don't realize that. They are thousands and thousands of miles away, come to us for cancer care, and also now have access in a way they have not before to these large kind of projects. And so this is a bit of just the demographics, and what I'll say that we're really happy about is that I have actually on multiple occasions pulled the 2020 US Census data, and this is again for US because that's the data we have. So this is limited to the 90% that are enrolled in the USA. This is the closest I've ever seen on anything we've tried to look, and we do a ton of work trying to increase equity inclusion on our clinical trials. This project kind of blows everything out of the water, being almost like the census data, which is great. This is actually kind of where things fall off. There are about 15% of kids who may enroll, but for one reason or another don't send the specimens in to be tested. And that may be that they didn't have they may not have had a biopsy at all. They may not have had a biopsy with enough tissue. They may have had a biopsy or had a tumor removed, left, didn't coordinate getting the blood sample sent. And you do have to have blood and tumor to have the testing start. But once that happens, there's only 5% that fail at what's called the Biopathology Center in Ohio, some sort of quality control. And the quality controls do include, are there enough tumor cells for methylation? There's a percentage that's required, so you're not studying the methylation of the normal cells. is there enough DNA? And there's a cutoff, which is about to go down with increasing technology. And then what's really kind of cool to see is when they pass those metrics, which again, 5% may have a tumor tissue issue, including not tumor on the specimen that gets sent. That's also, you fail if that's the case. There's a less than 1% failure in the DNA testing and about a 7% on RNA, which is much lower if people send frozen, but not all sites can even obtain frozen in their pathology departments. So it's a really high testing performance rate, which is the blue. And those are all done, these are all clinical tests, methylation, paired tumor normal whole exome, and RNA fusion panel, which are done at the Institute for Genomic Medicine, also in Ohio. And then the results go back to, the results don't belong to the COG, and that's actually confusing to a lot of people. So I don't have everybody's results. I am trying to get them through dbGaP. They go to the patient, and they go to the treating physician, basically. So they're returned like any send-out clinical test. They go back to the site. They can be used for anything, and they are used. We on the CNS committee of the COG rely on those for a lot of our studies. We've actually designed the studies in Parrot, knowing that people could have access to this testing. And then they also the data goes into the CCDI. So if you're a single patient, this is a great workaround for us not having a national healthcare system to get all your molecular testing clinically. But if you are a researcher, the data now belongs to all of us as part of the CCDI, and you can get it through dbGaP, and I'm not the expert at doing that. It took me 3 weeks to log into dbGaP, which is exactly how long it takes to get your exome results back, which I think is also ironic. This is one of the big things, and we heard a talk today about actually the Go Back project, which that group I know is going to go back and get this data as well. And one of the things I want to say is that so first of all, 12% of the kids tested so far have a germline, like with a very high bar to call this a clear germline cancer predisposition. But it's not any one gene. CHEK2 is actually really a common one, 53, but those are getting under 1% already. And so you actually have to look at everything if you want to get a complete picture. But this is clear, like clinically actionable information, which has relevance to patients and families. We're also excluding kids because you must have a newly diagnosed first tumor you've ever had. And that later part is excluding everybody who actually is pretty much known to have tumor predisposition or had a second tumor. So there are a lot of kids, like if you had Wilms tumor 10 years ago, we really want to know what that kid's biology is. They're not included in this because that wouldn't be their first tumor. This is actually what sites tell us. So there is one follow-up form for the MCI specifically, which is, was this useful? Did you use it? So did it change your diagnosis? And again, this is what the site CRA says, yes or no. Many of us may have different opinions on this, but this is sort of what the site's telling us. So 35% said it refined the diagnosis. And I think that's about like 15% said that it actually caused them to change or match therapy based on that, again, initial diagnosis. And then only 5% of those were enrolling on trials. And a lot of it is because, and I'll show you a little bit about what kind of tumors we're getting. The most common reason people wouldn't have been on a trial is there wasn't a trial for their diagnosis. So this, I think I'm missing one. This is just an example of, I think I got to sit next to Jay this morning. Medulla is probably one of the best characterized human tumors there is. And this is just a snapshot of what we have so far about medulloblastoma. We can clearly see by their methylation results what groups they fall into. The part that's most striking to me on this, and it's kind of as expected, it's a little highly represented for Wnt, and that is because we had a Wnt medullo study through COG, so I think there was a little impetus to enroll extra Wnt patients, even if you already had that diagnosis. But we got 412 patients in 20 months. We actually think that's about as many kids in the US that are diagnosed with medullo in a year. So we're getting probably more than half of the kids in the whole US already in the first few years. And this has been a ramp up. I think we're going to get well over half of the kids with CNS tumors enrolled probably in the next year. This is kind of how we use all three for medullo on the COG trial because you need methylation data, you need sequencing, and you need copy number information. And so all of that's used clinically. That's why we're getting such a good representative of medullo. A lot of sites you actually need this for clinical care, not just trials, and a lot of sites didn't have this. So it's a really mutually beneficial project, because we know kids in many of the hospitals didn't have access to the kind of data they need. And then I think I'm going to kind of stop here. The other thing that's actually been interesting to me, and I just took a look at this as an example of DMG, which of course are now defined by the H3K27 alteration. One, I'm surprised we got that many before dropping the DNA requirement, because these are often diagnosed by needle biopsies, but it's been actually remarkably successful. So there were 86 of these kids in the first 20 months. And I think there will be more once the DNA requirements go down. What was really interesting too, and I think a lot of us know this anecdotally, but maybe not with the looking in a larger data, is that they all have other mutations, which are actually, in this day and age, at least potentially much more alterable. This is sort of just this is done with the opposite of AI. I just literally went through the spreadsheet. But this is the example of the other types of mutations that we saw. And we actually have a trial open in the COG for BRAF mutant tumors, and we did see this overlap, which is rare, but you start to see it. And if you're contacting everyone, you can start to see it more frequently. Some of the other things are the things we don't normally look for, like CSF cytology, which we found positive in 14% of the kids that were looked at. We saw fusions in 7 kids. That is not a common test we run on this, but now makes me think we should be. Because it's just the lesson of we don't know what we don't know. So having a broad testing like this is really important. And 5 of those kids also had germline alterations that are quite relevant. Okay, so this has made me think a lot about how else we should approach clinical research in cancer in general. I think CBTN is really ideally positioned with the work we've been doing together to try to put together every type of data, to kind of take the lead on how and be an example of what kind of projects we could do now that we really didn't think about before, when we think about like identifying one gene, maybe one drug, maybe running a trial. I think not all the questions we need to answer are going to be answered through clinical trials. But the type of clinical research we can do with this data kind of blows my mind. And I actually think it will be people younger than me that will probably make the next big advances, and I hope to help support the infrastructure for everybody who's coming after. So I think that's it. What I have. I would really like to thank a lot of the people who put this together across really the NIH team, CCDI. It's been amazing to see just the enthusiasm for everybody working, especially kind of behind the scenes people. There are some folks at IGM who helped put together some of this data, the COG statistician team, including Yu Wang, who made me that lovely picture of the US. And then my partner, Diana Thomas, who's a pathologist at the Biopathology Center and IGM, who's been the main lead for the CNS project.
Just to, we did a lot of talking this morning about collaborating and connecting. And I think one of the really exciting things about CCDI that we didn't have time to put into the meeting is an initiative that talked about federating across a CBTN data set and an MCI data set. And one of the really exciting things is that really thanks to partnership with Sarah and the COG, approval of linking IDs between the COG and CBTN, with the NCI being the arbiter of connecting. We are now positioned to essentially link any MCI subjects data to a CBTN sample that Aaron Diaz might have done single cell on, for example. So I think that's one of the initiatives that we have never been able to do before, is to link across that context. And then longer term, I think there are opportunities to leverage lessons on the clinical data workflows, because as I mentioned earlier, and I think Sarah well knows, MCI is still human-based on the clinical data workflows.
Let's start with a question, and either one of you guys can answer. You hinted at this a little bit, Sarah, about how the data is ours, and yet for the common person, getting to that data still feels difficult. And I guess, what are the initiatives being done at the NCI level or the MCI level to really get the data into the hands of the physician scientists? Obviously, CBTN has done a huge amount of work. They're training people as we speak on this. But what about the data that comes into the CCDI? How does that get back to those of us who are researching?
Sure. So one of the things we're looking at is how do we get as much open data into things like CBio Portal? So that would actually be an unlocked CBio Portal anybody can use. And actually, I was taking notes this morning about can we rework our germline pipeline so that in fact, when we see 10 or more patients with a particular variant, we can make that available publicly. So there'd still be a screen for some mutations where we don't see them very often, and everybody who uses the resource would have to know that. Right now, if you go into the Genomic Data Commons and you look, all the germline mutations that are known to be germline and not just somatic mutations have been filtered. So we're thinking about ways to actually make a lot more data openly available. So again, that's separate from the whole discussion of should we be making all pediatric cancer data where we have clear consent from the families and from the patients that these data should be made freely available. Can we actually do that? That's a whole different to one of Adam's earlier comments. That's not a technical issue, it's a policy issue, and those move slowly.
Well, no, and I just would say, I think that the bar for getting the data is more frustration from those of us that don't know how to use anything beyond Excel. And I think we should talk openly about that, maybe not right at this moment, but I think if you're a researcher and you want to get the data and you heard some things in there, but you don't get it or you don't understand it, the older I get, the more comfortable I'm saying I have no idea what you're talking about. I say that to Adam all the time. I think it's really important to recognize that, like, we have different specialties. A lot of us in the CNS community are perfectly poised for this because I don't do neurosurgery and my patients depend on that for survival, right? I mean, like, we're already used to working in multidisciplinary. Being a data person is like being a doctor. It's not one thing. So there's a lot of other expertise that is not medicine and biology, where a lot of us come from. It's important for us to talk to each other and be honest. We need translation. Guess what? Diagnosis doesn't mean the same thing. One of the things I will say is great is we've got all the CNS data in the WHO CNS5 nomenclature in both CBTN now and on the MCI. And we're actually the first to do that. Like, they're still trying to figure out how to get the SEER data like that. So what we call diagnosis today is in there this way. And I think we're really, that's a unique opportunity that we've never had with the big data projects that we've tried to do before.
The entire day we've heard so much about AI and data. We have plenty of data. As Carl said, for simple people like us, it's hard to access those data. How far are we, or what does it make to have a chatbot that I could put my question to say how many such mutations are associated with such in DIPGs that are 4-year-old who have received such mutations, and get a graph for me so I can put in my proposal without having a bioinformatician on my team. What does it take?
Allison is going to answer your questions about that. That's
2 hours.
2 hours. 2 hours. She's really going to answer that question. That's a great question, and it goes back to how we make cancer data available. I will say that I'm pretty confident that generally NCI is quite aggressive about making data available, as aggressive as we can be. We want data available because we recognize exactly for what you said. It's really important if we want to make new discoveries. Data has to be available, but we also have to thread the policy needle. Right? So we're pushing pretty hard. I would love to see, and maybe Allison's going to talk about this, how we can connect some of these chatbots up to protected data and make sure that doesn't leak through those interfaces. I think there's some technical pieces there, but I'd love to see that happen.